Hello students, we have already studied this chapter, that is PTC Lab Organization, and PTC stands for Plant Tissue Culture Laboratory. It's a place where all sorts of plant tissue culture related activities, techniques, you can say methodologies are employed to grow or maintain plant cells, tissues or organs on a nutrient culture medium under aseptic growth conditions, and it has to be performed under controlled conditions. Now let us talk about basic facilities which are required. We require a washing area. We require where we will just wash the glassware, plasticware, where we'll keep the instruments, where we keep the media, and where we keep the chemicals. So we have area for media preparation also. It should be there, and transfer area where we'll transfer the material, and the culture room and the incubators. Now let us talk about in terms of general laboratory what should be provided with. We should be provided with the following areas. The washing area should be there, the area for media preparation separately, the area for inoculation. We have to perform this inoculation under aseptic conditions, and area for incubation because we have to go in for organogenesis. Shooting has to be carried out in light, and rooting is to be carried out in darkness. And also for cell culture and culture should be separately. Now let me show you one by one what are actually the requirements. First and foremost is the wash basin. You can see the wash basins. That should be neat and clean, well maintained, very clean area. You should have wash basin like this, running tap with running tap water, where you will just wash the material, etc. Running tap water should be continuous. Then we talk about the things which will be requiring for sterilization, this for media, etc. It is the autoclave here, and we also require a laminar flow, a horizontal type for plant tissue culture. Then we are just a lady who is working with the plates and with the plants. She's inoculating the things over there in laminar flow, and it's again observation of things under microscope while using the masks and aprons and head cap also. Head gear also can be used in order to avoid any type of contamination. It is here the media etc. is being prepared and kept ready, and bottles etc. you can see they will be filled with the chemicals etc. which will be required. From time to time we'll be preparing the media. We require macronutrients, micronutrients, we require vitamins, we require hormones, we require all things. We require sucrose also as a carbon source. We require agar-agar if we are going in for callus culture. If we are not going in for callus culture or cell culture, then we'll be not requiring the agar-agar. So all these things are there. Double distilled water, sterilized water, etc. is required for preparation purposes. So here you can see the well illuminated shooting area where the callus will be allowed to go in for organogenesis, for the shoot formation, and here you can see the root formation, and ultimately you can see the glass vials. It's actually the big bottles inside which the plants have been grown in for a plant tissue culture. Then we go for acclimatization and then hardening, and then we'll just plant them in the field. So media preparation room will have autoclave, accurate balance, pH meter, dispenser, 4 degree centigrade storage. We should have transfer room, clean room, laminar airflow, hoods, instruments and sterilizers can be used here. If you don't have laminar flow, you can use the hoods. And growth room should have temperature and humidity control. Horticultural lighting should be there. You know already I've shown you the well illuminated shooting area where the shoots are, where the callus is allowed to go in for shooting. And nursery, acclimatization and transplantation of rooted plantlets we'll do first with inverted beakers in order to reduce the rate of transpiration, but photosynthesis will be allowed first in the diffuse light, then in the bright sunlight, and ultimately hardening, and then we'll just allow them to grow in the field. Equipment and instruments required, we know already, balance for taking the more weight, and if we require less weight we use monopan balance, pH meter checking the pH, A digital pH meter is better suited, and containers, beakers, flasks, volumetric flasks, etc., volumetrics, pipettes, graduated cylinders. Other equipments, instruments include ovens, incubators, laminar hood flow, freezer to store samples. Last but not least, a big thank you to all my students. Please like it, share it, and subscribe. Thank you.